Hello everyone, welcome to your second PUC Biology practical sessions. So now let us begin with the first practical session. The aim of today's practical session is to learn about the different parts of a typical angiosperm flower. A typical angiosperm flower is bisexual and it has a stalk that is called the pedicel. Pedicel on one end is attached to the plant, on the other end it bears a flattened structure known as the thalamus. On the thalamus you can see 4 floral parts arranged in the form of concentric circle. Each concentric circle is called a whorl. So there are 4 whorls present on the thalamus. Let us learn about each one of them starting from the outside. The outermost whorl consists of structures that are small and leaf-like. They are known as sepals. Sepals, collectively, all the sepals together, is known as the calyx. Calyx's function is to protect the floral structures that are lying inside of it. The second whorl consists of brightly colored structures that are called the petals. Petals are collectively known as the corolla. The petals perform a very important function. Their function is to attract the pollinators. Inner to the petals is what lies the most important part in the flower. The third whorl consists of the male reproductive structure which is responsible for the formation of male gametes. This structure is called the stamen. As you can see, there are 4 stamens shown in this diagram. All the stamens together are collectively known as the androecium. Inner to the androecium, we have the last whorl. This whorl is called the gynoecium, and the gynoecium units are known as pistils. Now let us look at the individual structure of a stamen. Stamen is the male reproductive part of a flower, which essentially means that this part is what is responsible for the formation of male gametes. As you can see, there is a very long, thin, thread-like or a filament-like structure that is present here. This structure is known as the filament. On one end of the filament, you can find 2 knob-like structures. This is called the anther. Because there are 2 knobs here, the anther is said to be bilobed. The 2 lobes of the anther are connected by a sterile parenchymatous tissue that is known as the connective. When you take a transverse section of this anther and look at it under the microscope, this is what it would look like. This is one lobe of the anther and this is the second lobe of the anther. The 2 lobes of the anther are connected by a sterile parenchymatous tissue that is known as the connective. Within each lobe of the anther, you can find 2 regions where pollen grains will be synthesized. The regions or where the pollen grains are synthesized is known as the microsporangium. Since within this anther lobe there are 4 microsporangia, this anther is said to be tetrasporangiate. Now, the primary function of the anther is to produce pollen grains, and these pollen grains are going to be produced here. Because pollen grains are very important in the process of sexual reproduction, it has to be ensured that these tissues are very well protected. In order to protect the microsporangia within which the pollen grains will eventually be produced, there are walls surrounding it. Let us learn about these walls starting from the outside. The outermost layer in the wall is called the epidermis. If you remember, you learnt in 11th, any part of the plant, take the leaf or the stem, the outermost layer will be called the epidermis. So this epidermis serves a very important function of protection. And this is single layer. Underlying the epidermis, we have something called the endothecium. Endothecium also is single layer. Eventually, after the pollen grains are produced, they need to be dispersed. In order to ensure the dispersal of pollen grains that are produced here, these will help. Okay, so these endothecial cells will help in the dispersal of pollen grains. Inner to the endothecium, we have something called the middle layers. Middle layers may be single layered or they can go up to 3 layers. Inner to the middle layers, we have something called the tapetum. The tapetum performs the function of nourishment in the anther. So when the pollen grains here are being produced from the sporogenous tissue by meiosis, it is these cells that will nourish the developing pollen grains. Let us now learn about the female reproductive part of a flower called the pistil. A pistil has 3 parts. The top receptive part is known as the stigma. It is this part that receives the pollen from the anther after the pollen have been liberated. Following the stigma, you can find a thin, slender structure known as the style. At the base of the pistil, you can find a broad structure that is known as the ovary. Within the ovary, you can find the ovules. Now, based on the number of chambers that are present in the ovary, they are classified into multiple types. As you can see in this diagram here, this is a transverse section of the ovary. If you take a section of the ovary like this horizontally and look at it from above, this is how it would look. Okay? So within the ovary, you can find only one chamber. Because there's only one chamber found, it is said to be unilocular. Locule for chamber, uni for one. Because there's one locule, this kind of ovary is said to be unilocular. One good example for that is P. Here you can see there are two chambers. Since there are two chambers, this kind of ovary is said to be bilocular. One example for that is petunia. If it has three chambers, then it will be called trilocular. If there are four chambers or four locules, then it is called tetralocular. If it has five chambers, then it would be called pentalocular. The example for trilocular ovary is onion. For tetralocular, it is datura. For pentalocular, it is hibiscus. We will now learn about a very important concept, placentation. Placentation refers to the arrangement of ovules within the ovary. These are some of the types of placentation. The first one here is called marginal placentation. You may have seen the peas arranged in a pea pod, right? So how is it arranged? Within the ovary, you can see on one side a ridge is formed. On the ridge, you can find the ovules attached. The ovules eventually become the seeds, right? Ovules itself will develop into the seeds. Since these ovules are arranged in the margin or on the margin, this kind of placentation is said to be marginal placentation. The second type of placentation is called axial placentation. Axial refers to axis. As you can see in this ovary, there is a central axis and it is multilocular. These are the ovules. The ovules are attached to the central axis, due to which it is known as axial placentation. A very good example of this is the hibiscus flower. Third, we have parietal placentation. Parietal means the wall of any empty cavity or chamber. So parietal placentation would mean that the ovules are attached to the wall of the ovary. So this ovary is unilocular. It has only a single chamber, and to the unilocular ovary's walls, you can find the ovules attached. The fourth one is called free central placentation. If you look at this diagram, it is kind of similar to axial placentation, but the difference is that this does not have a central axis. Here it is multilocular, here there is a single locule. The ovary has a single chamber and the ovules are attached in the center, due to which it is called free central placentation. One example of this is chili. Okay, the last one is called basal placentation. In the first four types, these diagrams are transverse sections of the ovary. That means the ovary, the sections are taken horizontally. For this one, the ovary section is vertical. There is a single chamber in the ovary, as you can see, and the ovule is attached to the base, due to which it is called basal placentation. One example for this is sunflower. What I am going to perform right now is one of the experiments that may be asked in your final practical exams. The experiment is called transverse section of the ovary. What I will need to perform this experiment is a hibiscus flower, a sharp blade, a slide, forceps, a brush. First, I am going to add a drop of water on the slide. First, you have to take the hibiscus flower and hold on to it by its pedicel. If you let the pedicel get cut, then it becomes difficult to take a section. So, while holding on to the pedicel, remove all other floral whorls either with the help of your fingers or with the forceps. So right now I am removing the green part, which is called the calyx. Next I will be removing all the petals. I have even removed the androecium. What you can see here is the pistil. This is the stigma, this is the style, and this bottom broad portion is called the ovary. And now I am going to take a section of this ovary. As you can see, the ovary is not uniformly round. It is a little narrower at the tip and a little broader at the base. In order to get a neat section, it's advisable that you take the sections from the center, because here it is wider and you will get a clearer section to show to the examiner. Now I am going to remove all other parts of the pistil and retain only the ovary. With the help of a new sharp blade, I am going to start taking sections of the ovary horizontally. These horizontal sections are called transverse sections. You can take many thin sections. You will have to show only one to the examiner. You can take as many as 4 or 5 and show the best one to the examiner. So this we are performing to observe how many locules the ovary has and also what kind of placentation hibiscus shows. So right now I have with me many sections, of which I am going to pick the best one. I am going to put it on the drop of water that I have taken in this slide so that I can examine them better. I have taken around 6 sections here, of which I am looking for the thinnest and the one in which the locules are very clearly visible. I am now going to focus the best section under the dissection microscope. What you need to observe here is the number of locules in the ovary. I can see 5 locules, so this is a pentalocular ovary. Also, the ovules are attached to the axis in the center, so the placentation is axile. The experiment that we performed today carries 5 marks in your practical examinations. 3 marks are for performing the experiment, whereas 2 marks are for reporting the result. The result needs to be reported in the form of 2 statements, each of which will carry 1 mark. The first statement, you have to write that the placentation in the given specimen is axial placentation. The second statement, you will have to write the number of locules observed in the given specimen's ovary is 5, therefore it is a pentalocular ovary.